And thanks for everyone joining online. Delighted to share the story of Argenica Therapeutics for those that are new to seeing what our company has to offer. So next slide, please. So just a disclaimer on forward-looking statements. Next slide. So just to give you a bit of background to Argenica, what we're aiming to do and why we're trying to do it. So we are a clinical stage drug development company, and we're looking to develop novel neuroprotective drugs or therapies that are able to protect brain cells following injury. So as Tim mentioned, we're looking at stroke, but also other types of brain injuries such as traumatic brain injury, Alzheimer's disease. And really the reason that we're doing this is ultimately to positively impact the lives of these patients that really have no option in terms of treatments at the moment for protecting brain cells. And in doing that, we are unlocking this huge commercial opportunity. So all the indications that we're going after, all those brain neurological conditions, they are large unmet medical needs that are global problems. Next slide, please. So just to give you a bit of a snapshot on our company metrics as we stand at the moment. So as of last quarter, 31 December, we had just a little over 8 million cash in the bank. Since then, we've also received around 3.7 million in grants, and that is predominantly to progress our preclinical work in traumatic brain injury and hypoxic ischemic encephalopathy. We're sitting around, as Tim said, that sort of 35-36 mil market cap, but we've had a pretty strong share price performance since IPOing in June 21 at 20 cents. Next slide, please. So just to give you an idea of our lead drug candidate, why we think it's really exciting, why we think it has a huge amount of promise, not only in stroke, but other neurological conditions. So the drug is known as ARG007. It has been in over 10 years of development out of the Perrin Neuroscience Research Institute and the University of WA here in Perth. It has over 25 peer-reviewed publications, not only on the efficacy of the drug, but also the safety of the drug and how it's actually working in the brain. We have recently completed a phase 1 clinical trial, which I'll touch on a little later in the presentation. The company does own all the IP outright and is free of any encumbrances, and we do have patents granted in multiple jurisdictions globally, including the US, Europe, China, and Japan. Next slide, please. So why are we looking at stroke as the first indication to progress into the clinic? Stroke is a huge unmet medical need. So 1 in 4 people will have a stroke in their lifetime. That equates to about someone in Australia having a stroke every 19 minutes. And of those people that sustain a stroke, only around 10% of them will fully recover. And that's because of the extent of brain injury, the neuronal cell death that you see post-stroke. And there are currently no drugs universally available that are able to protect brain cells after stroke. So this is the market that we are going after. And we think that we have a pretty good chance of being a leading company in this space. Next slide, please. So to give you a sense of how the drug actually works, so we are looking at ischemic stroke. An ischemic stroke is a stroke caused by a clot in a blood vessel in the brain. So 85% of all strokes are ischemic in nature. So what happens when you get a blood clot in a blood vessel in the brain is it blocks blood flow to that part of the brain, depriving the brain in that area of oxygen. Neurons or brain cells require oxygen to survive, and they can't survive very long without oxygen, only a matter of seconds. So once one neuron or one brain cell is deprived of oxygen, it dies and it sets off this cascade of cell death within the brain. So you get this proliferation out of cell death over time. And there is a saying in stroke that time is brain. So for every minute that you're delaying the removal of that clot, you're getting this cascading or this increasing amount of cell death in the brain. So what our drug does, it actually slows that progression of that cascade out of neuronal cell death. It almost hibernates the neurons or the brain cells, shutting them down and preventing that brain cell death, buying that patient time to get to the hospital. Next slide, please. So if I look at around how our drug would work in current standard of care, so currently when someone has a stroke and the paramedic is called, there's very little that the paramedic can do in terms of triaging that patient. The idea is to get that patient to the hospital as soon as practically possible, because doctors need to be able to determine or diagnose the type of stroke. So if it's ischemic, then they need to be able to locate where the clot is in the brain and how the treatment of that clot will progress. So that's either removing the clot through a non-invasive procedure undertaken by a radiologist, in which they put a device into the brain to pull the clot out, or they are given a drug which dissolves the clot. But that takes time. So all the time from getting in the ambulance to getting a scan to diagnose the type of clot to working out the treatment plan, you're still getting that cascade out of cell death in the brain. So what we're aiming to do is to be able to deliver our drug to patients as soon as possible, because it doesn't matter the type of stroke a patient has. And that will buy that patient time, shutting down those neurons, protecting them from that neuronal cell death, and buy that patient time to get to the hospital and get the treatments they need. Next slide, please. So what we've shown in a primate model, in which then this data has been published in a peer-reviewed journal, is that our drug is able to reduce that total volume of cell death, what's known as the infarct area or infarct volume, by 66% 24 hours post-stroke. And we can see that that effect lasts out 28 days post-stroke. So even though it's only a single IV injection, we are able to protect that brain from that brain cell death out to 28 days. Next slide, please. So we have really encouraging results today. As I said, 70% reduction in brain cell death, 28 days post-stroke. We also know that it is safe to administer in the field regardless of the type of stroke. So as I mentioned before, the clot in the brain, the ischemic stroke, that is the main type of stroke. The other type is hemorrhagic stroke, which is a bleed on the brain. We know that our drug doesn't exacerbate bleeding. That's really important. And then we also know that we don't have any side effects in the phase 1. Next slide, please. So just on our phase 1 trial, so this is done in healthy volunteers, and what we showed was that our drug does not induce any clinically significant adverse events. It was completely safe, it was well tolerated, and in fact we saw more mild adverse events in the placebo group than we did with the treatment group. So this gives us the confidence to move into a phase 2 clinical trial in stroke patients. Next slide, please. So just in terms of the timeline for the phase 2 clinical trial, we're hoping to be able to dose patients coming into the emergency department that have had an ischemic stroke by the end of this calendar year. There's a few things we need to do to get there. So the main thing being submitting an ethics application to an ethics committee to get approval to undertake that phase 2 clinical trial. That clinical trial will be done solely in Australia at stroke care units in most major cities across Australia. Next slide, please. But we do see ARG-007 very much as a platform technology. So whilst we're going after stroke, that's where the majority of our preclinical data comes from. There are a raft of other indications that we are looking at. So the most advanced are the hypoxic ischemic encephalopathy. This is a reduction in blood flow in newborn infants, and it's usually caused by the cord around the neck or preeclampsia in the mother, things like that, where you're preventing blood flow to the brain in these infants. And the consequences following this reduction in blood flow in these babies is dire. So it can result in severe disabilities, including cerebral palsy, but also learning difficulties and motor function disabilities. So we've got some really positive preclinical data in HIE, and we're also progressing into severe and mild TBI relating to concussion. And we've had some positive preclinical data in Alzheimer's disease. Next slide, please. So as I mentioned, stroke is the main focus for the company as we progress into the clinic, but we are also progressing into setting up all the studies that we need to do to progress into the clinic for HIE. So recent data, we show that the drug has an 86% reduction in that infarct volume when a single dose is delivered following HIE. We also have really encouraging data in traumatic brain injury. And as I mentioned before, we've just received a grant for $1.2 million from the federal government to progress preclinical studies in concussion, which is really exciting. Next slide, please. And then encouragingly, we've also recently showed that our drug is able to significantly reduce beta amyloid aggregation. So this is one of the key hallmarks of Alzheimer's disease. So the drug was reducing the amount of amyloid aggregation that we see in a cell culture by more than 50%. So we're really keen to progress these preclinical studies and work out how effective our drug is in an animal model of Alzheimer's disease. And we've received some philanthropic funding to progress that as well. Next slide, please. So along with myself, I am supported by a fantastic team, which has not only capital markets experience, but experience taking drugs all the way through to FDA approval. We're really excited about the next stage of development. We're excited about getting into the clinic. I've recently come back from a trip to Washington and having discussions with the FDA around, how we can accelerate getting some of these drugs to market. So really exciting, great to be working with the FDA and lots of positive things to come. Next slide, please. So just to quickly recap, I can see Tim on screen, so we'll race through this. We do have a really solid foundation of preclinical data, which is a really key aspect of de-risking this asset. So not only showing efficacy, but safety, tolerability, and mechanism of action of the drug. Strong balance sheet. We've shown that it's safe in healthy volunteers, which is a huge hurdle to get across in drug development. And really exciting next sort of 12 to 18 months coming up. So thank you.
Thanks, Liz. Fascinating story in what is some very important work. I've got a couple of friends who, in the 40s, maybe early 50s, who've had strokes. And one of the issues around strokes is there's some telltale early signs. Can't raise an arm, I believe, or some of those signs. Is this almost preemptive? Can the drug work best when you get the stroke patient early?
It will work at its best, if we can administer pretty soon after stroke onset, because what we want to do is we want to prevent that cascading out of cell death. So yeah, absolutely, the sooner the better. One area that I didn't touch on that we're quite keen to explore is what's known as surgically induced stroke. So that's when someone goes in for heart surgery, and around 10% of those patients can actually have a stroke during that surgery. And that's just in terms of dislodging clots that go up to the brain during that surgery. And so what we would hope to do is be able to deliver our drug post-surgery, almost shutting down the neurons in the brain, protecting the brain as that patient goes in for surgery. And that's preemptive, right? So then if they do sustain a stroke, at least the neurons are protected.
Certainly makes sense. And what does the phase 2 trial look like?
Yeah, so we are just in the process of finalising the protocol for that, but that will be, I guess one of the key points to note is a lot of drugs fail because of bad clinical trial design. So getting the trial design right is really, really critical to the success of sort of progressing the drug through. So we will be delivering it in the emergency department. That's because we want to know exactly the location of the clot. And the reason for that is to reduce the amount of variability, because strokes can be quite variable, and we do want to get some sort of efficacy readout. So we want to see some sort of functional recovery of these patients. So if we can reduce the variability in where the clot is, the sort of size of the clot and the size of that infarct, then we'll get a better chance of actually seeing some kind of efficacy of the drug. So patients will come in, they'll be scanned, and then they'll be brought onto the trial, deliver the drug, and then they'll go into for their treatment to get the either the clot removed or the clot dissolving drug.
And of course, you're well funded in what has been a very difficult market for biotechs. Where are the funds going to be used, given you've got multiple applications?
Yeah, so look, the majority of the funds really are for the stroke that we've got in terms of cash on hand. We are lucky that we have now got close to $4 million in non-dilutive grant funding. And so those grant funding amounts are for HIE and TBI. And we've also now got philanthropic funding for our Alzheimer's. So we've been able to get all the non-dilutive funding to progress the other preclinical indications. So the cash that we've got at the moment will focus largely on the stroke trial.
And is there any similar question here? Is there any similar sort of products in the market, potentially in the products or drugs, sorry?
Yes, there are. So there is not in the market. There is a drug called NA1, which has just finished a second phase 3 clinical trial. So that's a global clinical trial. Recently met with the founder in Washington as well of that company. And so that is quite a similar drug. So it's neuroprotective, but it cannot work with the clot dissolving drug. So it's targeting sort of a narrower subset of stroke patients. But fantastic. It's always good to be a fast follower in these sorts of instances. If they get their drug to market, it'll probably take around 3 years for that for neuroprotective drug to be reimbursed in the US. So we're very happy for them to pave that pathway, and then we can come in. And I think generally the feeling amongst clinicians, neurologists, the FDA is that neuroprotection will be a little bit like oncology. So there'll be a number of neuroprotective drugs on the market, depending on the type of stroke, when that patient's got to hospital, the location of the stroke, and what the treatment plan is. So there will be a number. There's not many in the clinic, I have to say. So we are sort of hot on the heels of the first 1 or 2 that will get to market.
Yeah, very well positioned. And just to finish up, Liz, can you talk through the kind of milestones you've got to achieve over the next 12 to 18 months?
Yeah, sure. So the key, we need to get the final clinical trial report from the phase 1 clinical trial, and that will just show that the drug's safe, it's effective, and then that goes into our ethics submission. So we submit our ethics to a national ethics committee. That takes around 3 months to get that approval, and then we start setting up all the sites and dosing first patient at the end of this calendar year. Dosing will take around 18 months, so that whole trial will probably take around 2 years. We'll have locks in between, plenty of stuff to announce throughout that trial, especially around the safety of the drug.
Fascinating story, Liz. Thank you for your time. Have a nice weekend.
Thanks, Tim. You too.